So we are talking about genetics. Genetics has to do with the way genes are passed from offsprings to, not from offspring, but to from parents to offsprings. Just the way genes are going to be passed. It is as easy as that. So when we are talking about genetics, let's say for example, my mother, let's say I've got my mother here, and then we have got dad there. Okay, so let's say my mother is, let's say, tall, and then say my father is not tall, let's say is short. Yeah. Okay, forgive me for that. It's just a camera board I'm using, that is, let me just connect it well. Okay, so let's say the father is short, and then you find that after crossing the, you find that the offsprings are, let's say all of them are tall. So this should tell you something. This tells you that the mother in this case has got a dominant characteristic. What we mean by dominant is a characteristic that is going to influence, or that is going to determine how the offspring is going to be. So this is going to be the offspring. Okay. So in genetics, we are going to have first a physical appearance. Like as you can see, you can see that the offsprings are tall. That's the physical appearance. The physical appearance is known as a phenotype. Phenotype. So what is a phenotype? It is an observable physical characteristic. You see it. It is observable. Okay, not something that you can see. For example, you are tall. You can observe. Observable physical characteristic or trait. That is the phenotype. But then for you to be tall or for you to be short, you are made up of genes. So those genes are going to tell you something. So your genetic makeup is known as your genotype. So what is genotype? It is an individual's collection of genes. An individual's collection of genes. Now, for your genetic makeup, you don't only have one gene. Okay, you are going to have a gene, but then information is going to be coming from your mother and from your father. So each gene is going to be made up by alleles. Okay, so gene is equal to allele plus allele. And one allele is from the father, another allele is from the mother. So we define an allele as the alternative form of a gene, another form. Okay, because the gene is going to have two forms, and the gene specifies the kind of trait that you are going to see. Now, for example, from your father, you can get an allele. Let's say, let T, capital T, be for being tall, and then let's say small t being for short. When you see small letters, it represents a recessive trait, meaning that it's a trait that you not see. It's going to be overshadowed. A capital represents a dominant trait. Let's say, for example, in the example that I gave, the father is short. It is TT. The mother is tall. Capital T, capital T. So these are the genetic makeups of the mother and father. But then as alleles, allele is a gene has got two things. A gene is going to have two alleles. That's a gene. But an allele is an alternative form. Just one of these t's that makes an allele. Let's say from my father, I got a small t. That was the allele I got. From my mother, I got a capital T. So when I combine this, because I've got a capital T and a small t, I'm going to be tall because big T is capital. But if someone has got both t's being small, for such a one, they are going to be short because they only have recessive alleles. So we need to also understand what we call homozygous and heterozygous. Okay, or homozygote and heterozygote. An organism that has the same two copies of a gene is homozygous. So that is the same alleles. That's homozygous. For example, someone has capital T, that's homozygous. And then someone that is a heterozygous, or a heterozygote, this one is going to have different forms of a particular gene, okay, or alleles from each parent. Different alleles. An example that we can give, someone has got capital T and small t. Now, when you look at alleles, there is always one allele that is dominant over the other. So that takes us to what we call dominance. So what really is dominance? So dominance is having greater influence, or one allele having greater influence on the other. Greater influence over the other. Okay, so that is what we call as dominance. And these two guys, they specify the same trait. For example, if I'm talking about the color of the eyes, I'll say one allele is dominant over the other because let's say my mother is saying I should have brown eyes, my father is saying I should have blue eyes. The one that is dominant will determine what kind of eyes I have. So alleles having greater influence over the other for the same trait. Okay, that is what we are calling as dominance. So having understood this, we also need to know there are different types of dominance. There are different types, and there are three of them that we need to understand. And I'm going to write it on top here. Please understand this well because it's coming, all right? What are the types of dominance? We have said we have got three types of dominance. The first type is known as complete dominance. The other is known as codominance. And then the other one is known as incomplete dominance. So you should be able to differentiate this. Now, how we are going to explain all these traits, let me just move this a bit so that I can have a small space here. So here we're going to have complete. Okay. So now let's say we're talking about alleles. Let's say we're talking about alleles. Let's say, for example, we are talking about color. Color. So let's say we are going to say let capital R be, say, okay, let me use, what letter can I use? Okay, let capital R be red. And then let say, let me write them in their respective color so that it can just, and even just like this. So let capital R be red, and then let's say small r be black. Okay, there. Now let me swap them because black is supposed to be dominant. Remember, when you use capital, it means that that trait is dominant. If we use small letter, that trait is not dominant, it is a recessive. So this tells you that if I have got capital R, all right, and another capital R, because capital R is representing uh, black. So if I've got two R's, it means that whatsoever the organism that we have is going to be black, whether it's complete dominance, incomplete dominance, or whatsoever kind of dominance here, because you are using the same allele and that allele is dominant. Okay. And then if I have got both being small r's, it means that that organism is going to be red. Only small. Remember, the small is for red. It is recessive. But imagine I've got a combination of capital R and small r. Then what is going to be the result? In a complete dominance, it means that one allele is going to completely overshadow the other one. That's the meaning of complete dominance. So because the capital R is dominant and capital R is representing black color, it means that the organism is going to be black. Now, in co-dominance, co means that the two traits are going to appear at the same time. That's the meaning of co. Co means working together. So you have got an area for black color, an area for red color. So if they come together in co-dominance, instead of one of the other, the animal that you are going to have is going to be both black and red. Is going to have patches which are black, some patches are red. But in incomplete dominance, when you combine one allele with another allele, this is incomplete. Like one is supposed to dominate over the other, but not completely. Think of it, you get juice which is red, you get juice that is, let's say, green. You combine them, the color that is going to be formed is going to be something on the middle. So when you combine in incomplete dominance, you get something that has got a black color and a red color. You are going to get a color that is like in between, something like purple. Okay. So you are getting a color that is in between, that is incomplete dominance. Does this make sense? Let's respond so that we get to move quickly. Okay, so that is about dominance. Okay, so we have got dominant genes and recessive genes. Now, what is a recessive gene? When you hear recessive, it is a gene that is going to be overshadowed when it comes uh, to having a characteristic. So a recessive gene, this is a gene, okay, recessive allele, specifically because it's not a gene. A gene is a combination of alleles. So a recessive allele is an allele which is masked, which is masked. To be masked means to be covered. You can't see it if they come together with a dominant gene if it comes together with a dominant gene. And you use a small letter for this. Use a small letter. For dominant guys, you use capital letters. Let's give an example. Okay? An example. Let's say you have got your mother. We are going to talk about first the genotype. Okay, we start with the phenotype. Phenotype is a physical appearance. Phenotype. And what characteristic can I talk about? I can talk about height. Okay. Let's say you have got a tall mother. You are crossing it with a short father. Now, the thing is that when someone is tall, you don't know the genotype. And these are the questions that they are going to ask you in the exam. You can determine. So if someone is tall, they can either be capital T, or capital T and small t. Remember, as long as you have got one dominant allele, the appearance is going to be always dominant. Let's say it is dominant heterozygous. Heterozygous means you have got capital and small, and you are crossing this someone that is short. Short is always both are small. So what happens is that before these guys can come together, you are going to have gametes. The gametes, these are the ovaries and the sperms. So the ovaries and the sperms, the gamete, each one of the gametes will get one allele. That's why we say that the gametes are haploid, because they only contain half the number of chromosomes. And then now these guys will be able to cross. When they cross, what you are going to see is this capital T will cross with this capital T. So we are going to have T, T. This capital T again will cross with this capital T this side. So we are going to have capital T, small t, and then you are going to have this one crossing with this one. So be small t, and then you are going to have this one crossing with this one. So it's going to be small t, small t. So what you can see, because this is showing the genes, this is known as the F1. F means filial. Okay. So this is known as first filial genotype. F1 genotype. You might remember from your biology in high school. So from the F1 genotype, now you can determine the F1 phenotype. Remember, the phenotype is a physical appearance. So when you have got a capital T, that means that this is tall. A small t, there's still a capital A, that's tall. These are only small, that is short. This one is also short. Now, what you can see here is a probability that when this woman and this man come together, the probability that they are going to have a tall child. Please, it doesn't mean that when they have got 4 children, 2 are going to be tall, 2 are going to be short. No. This is telling you that look at a tall one. How many tall do you have? You have got 2. So the probability of having a tall child is 2 out of how many children? 4 times 100. That is 50%. In other words, when a child is born, when the woman is pregnant, there is 50% chance that that child can either be tall or short. Not that when they have got 4 children, 2 of them are going to be tall. So each one of the children that this couple is going to have, even if the children are 50, each one of them has got a 50% chance of being tall and 50% chance of being short. And it is possible that all the children can actually be short because each child had an equal chance of being tall or short. Okay, so hope we have gotten that. So in genetics, we have got also what we call a test cross and a back cross. So what do these mean? Starting with a test cross. A test cross is a cross between an individual with an unknown genotype and a recessive parent. Please take note. Okay. So cross between individual with an unknown. Please take note of that. An unknown genotype, not phenotype. Genotype. You know their phenotype. Sometimes. Okay, but obviously, phenotype is always known because you can see if someone is tall, if someone is short, if someone is dark, if someone is light. So, cross between individual with unknown genotype with a recessive parent. Recessive parent. If not parent, at least someone that is also similar to the parent. Now, if someone is recessive, it means that, let's say, for example, if you're talking about recessive to height. Someone that is recessive to height means that they are short because it is small t, small t. Someone is recessive, let's say, to color. Let's say someone who is dominant is dark. It means that someone that is recessive is going to be light. Okay, so that's the meaning. So what you are trying to do is that you, what is the aim of this test? It is used to determine whether an individual is homozygous or heterozygous. That's a use. And this is how they are going to ask you in the exam. Okay, what is the test, or they can ask you what is the use. So it is to determine if an individual is heterozygous or homozygous. Remember, for example, you know that someone is tall. But I said that if someone is tall, they can have a capital T and another capital T, or a capital T and a small t. So you can't really determine which genotype they have. You already know their phenotype, you know their appearance physically. They are dominant, but then you don't know if they are dominant recessive or if they are dominant homozygous or dominant heterozygous. Let's do an example. Okay. So in this example, say we have got we have got I can write it here. I can say Mwanza is tall and is crossed. Okay, there is no need of me even mentioning who I'm crossing with. Remember, we are performing a test cross. Immediately you hear test cross, it means that what you are crossing with is someone that is recessive short. Okay. So what is important is, let's say a tall flower is crossed in a test cross, and all the resulting flowers at all. What is its genotype? You can see the kind of question here. Okay. So how do you answer this? You basically perform, you can perform a cross using the one I gave you, or you can use a simple one where you use the grid like this. Using a grid is even much simpler. Okay, there. And here, remember, we are crossing, we are performing a test cross. And because this is a test cross, it means that we are using another flower that is small t, small t. But this is the genotype. When you are performing a cross, you use the gamete, so you split those alleles. So I'm going to put one allele here and then another allele here. Now remember, we have been told that all the offsprings are tau. So this is going to be tau, this is going to be tau, this is going to be tau, this is also going to be tau. So if all the offsprings are tau, already you can see that from this t, this small t and this small t, at least in each one of these boxes, you're supposed to have a small t. Okay? Like below each t, below this one supposed to have a small t, even below this one a small t, coming from this t, and then from this one a small t, a small t. Now the only way the offspring can be, all of them can be tau, is that because you already have one small t, it means that you're supposed to have a capital T here. Also you're supposed to have a capital T. Also you're supposed to have a capital T. Also here. This is telling you that the allele that we had here was a capital T. The allele that we had here was a capital T. That's the meaning. So already you can determine, you can see I've got two capital T's, which are the alleles. It means that the offspring was actually capital T, capital T. Do we get this? This is the genotype of the flower. So we started with the offsprings that we had, and then we were going in the backward direction, and then we determined the offspring of the one that we used to cross. Okay, hope that is clear. Now imagine if we are told that not all the offsprings were tau. Let's say only 50% were tau. If only 50% were tau, it means that the other ones would be short. So in that case, what we would have done, it would be something like this. Remember, only 50% are tau. So it means, and in a test cross, always one of the participants is always having recessive. So if 50% are tau, let's say for example, this is a T, because all these are small t's, right? So at least in each one of these boxes supposed to have a small t. So if 50% are tau, we can have a capital T here. So a capital T is going to come here, capital T will come here. So meaning that these are tau. But what about the other, which are the 50% which is not tau? That is going to be a small t again coming from the other parent. So this is going to be here, small t, small t. So this is telling you that this 50% which is down is short. The 50% here is tall, because every time you have got small t, that is short. So it means that the 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 unknown genotype would be capital T, small t. Have I made this clear, or someone is lost? Okay, I'm guessing we are good. So this is about a test cross. Now, how about a back cross? So what happens in a back cross is we are getting to mate hybrid organism with one of its parents. Okay, not just the parent that is recessive, but mating hybrid organism, okay, with one of its parents. If you don't want to use the parents, at least use an organism that has got the same genetics as one of the parents. Okay, organism. So organism with similar genetic makeup to the parent. Please take note of the definition for the multiple choice questions. Okay, so this is what we call a back cross. And the reason as to why we even use a back cross is for genetic. Why is it important anyway to perform a back cross? Back cross is used in the genetic studies for isolating certain characteristics in a related group of animals. Okay, so we can give an example. Can give an example. Let's say, for example, we have got an offspring. Let's say we started with the parents. Uh, a parent has got the offspring has got, let's say, the genotype of the offspring is T, capital T, small t, and then one of the parents, the genotype is also capital T, small t. So now you are crossing these guys. So basically what you do is you can see from the offspring you have got capital T, small t, and then from the parent capital T, small t. So the offsprings that you are going to have in this case is going to be capital T, capital T, capital T, small t, capital T, small t, small t, small t. So basically that is a back cross. Please don't forget that you are getting the hybrid. Then you are crossing it with one of the parents, any one of the parents. So another thing that you need to understand are just the three laws of the Mendel's laws. Okay, laws of Mendel. So Mendel proposed the three laws for genetics that we have to understand. They are also known as the Mendelian laws. They are important for you to understand if you are to understand how characteristics are going to be passed from offsprings to from parents to offsprings. So the first law is known as the law of segregation, and the law of segregation simply tells us that when you are forming gametes, the alleles for each gamete will segregate from each other such that each gamete forms carries only one of the alleles. Like you saw earlier when I wrote like this, we had capital T, capital T. So when you are forming gametes, each gamete is only going to carry one allele. That's a law of segregation. And then the second law is known as the law of independent assortment. Law of independent assortment. And this law tells us that genes for different traits assort independently. This is an important law. Okay? Genes for different traits assort independently. It's actually this same law that we are going to prove by what we call the chi-square table, independently. So what does this mean? Let's say this is a chromosome. At one end of the chromosome, you have got, let's say here, say you have got eye color. And say at this end of the chromosome, you have got, say, height. So if this is the what you are getting from your mother or from your father, when you are forming, when assortment is happening, when you are forming gametes, law of independent assortment tells you that it is not that every time you have got a blue eye color you are going to be tall, or every time you have got brown eyes you are going to be short. These characteristics assort independently. You can have blue eyes but you are tall, or blue eyes and you are short. That's the meaning of independent assortment. Please take note, right? And then the third law is known as the law of dominance. And what does the law of dominance tell us? So the law of dominance tells us that when two different alleles are present, only one is dominant and will be expressed. When two alleles are present, only the one dominant is expressed. Now, this law, you can see it has got limitations because remember earlier we talked about codominance, where both alleles, okay, both alleles can be dominant. And when both alleles are dominant, it means you are going to undergo what we call codominance or incomplete dominance. But this law says that only one allele is dominant. Okay, only one allele is dominant and is expressed. Okay, hope that is fine. Please, this is very vital for you to understand. Let's just do some crosses before we can finish today's class. So we can give an example. Say you have got a man that is heterozygous. I can write that a man is heterozygous for, let's say, brown eyes. A man is heterozygous for brown eyes. Okay, and he mates say with a woman who is also heterozygous, also heterozygous. What is the percentage of their children having blue eyes? What is the percentage of their children having blue eyes? So already from what we can see here, okay, already from what we can see, both parents are heterozygous for brown eyes. Now let's say capital. Which one can we give capital? Okay, at this point, are we able to determine? Okay, since it look, they have said heterozygous and then it is brown. So meaning that, let's say we are using B to represent color. So they are both BB like this. So every time someone is heterozygous and then they have got a characteristic, it means that that characteristic is a dominant characteristic. So you're having two alleles, but then you have brown eyes. It means that brown eyes are actually dominant. Okay. So you can put that table that we gave earlier. So remember, both parents are heterozygous, meaning that both have got a capital B and a small b. Even the other one capital B and a small b. So of the offsprings that you are going to have, you are going to have a capital B, a capital B. This will be capital B and small b. This is going to be a capital B and a small b. This will be small b and small b. So you can also find what we call the genotypic ratio. Genotypic ratio, just looking at the genotypes. You can see capital B, we only have one. And then capital B, small b, we have got two of them. And then small b, we have got one. So this is a normal genotypic ratio for a monohybrid cross, 1 to 2 to 1. You are going to see it in exam questions. But what will be the phenotype? The capital B and capital B means they are going to have brown eyes, because remember, as long as you have got a capital B, it means you have brown eyes. Even when you have only one capital B, still brown eyes. So these two are brown eyes. And then when you are just having small, that will now be the blue eyes, they've said. So you have got the three blue brown eyes and one blue eyes. So the genotype, the phenotype, the physical characteristics, the phenotypic ratio is going to be 3 to 1, because you have got the three which are brown eyes and then one that is blue eyes. So this is the normal phenotypic ratio for a monohybrid. So now what is the percentage of their children having blue eyes? You can see blue is only 1, and the total is 3 plus 1, that is the total is 4. So multiply that by 100%. So the chance of having blue eyes is going to be 25%. Okay, that is how you calculate this. So let's just look at some questions before you can conclude. So I want you to understand something as we get to finish today's class. When you are performing a monohybrid cross, normal normal monohybrid cross. Please take note of this, is very important. The normal one. How does it go? So for the normal, we are starting with, you are first going to cross two capital letters with two small letters. This capital crosses with this small, you are going to have capital T, small t. This capital again with this small, capital T, small t. Again this capital T, small t. Again this capital T, small t. So the offsprings that you have gotten here, because what's on top, these are the parents. Then what you have gotten here, we call them as the F1 generation. And this is a genotype of the F1. And then now what you are going to do is that you get one of the F1 offsprings. And you can see all of them have got the same distribution, right? So I'm going to get a capital T, small t, and then a capital T, small t. Get any two of the offsprings. They'll now be acting as a parents, the F1 genotype generation. So this will cross with this to be capital T, capital T. This will cross with this, capital T, small t. This will cross with this, T, T, and this will cross with this, small t, small t. These other children which have been produced by the children, we call this as F2 generation. Their genotype is known as F2 genotype. So when you look at this genotype, the genotypic ratio, okay, this is important, which is the normal genotypic ratio for a monohybrid. Genotypic ratio. Can you see that we have got capital T, and then we have got capital T and small t. We also have small t, small t. Capital T, we have got only one. Capital T, small t, we have got two. And then small t, we have got one. So this is the normal genotypic ratio for a monohybrid. And then phenotypic ratio. Let's say T is for height. So our men are going to be tall. So we're looking at the ratio of tall to those which are short. As long as you have got one capital T, it means you are tall. So the first one is as the capital T, the second one and the third one. So that is the 3 to 1. This is the normal phenotypic ratio for a monohybrid. And then this is the normal genotypic ratio for a monohybrid. This is important to know. So we can now go on and answer the questions. In a monohybrid cross, the recessive phenotype disappears. Can you see what is happening? What is happening to the recessive phenotype? Is it disappearing? It is still there, all right? It is still there. Recessive phenotype disappears in the F1. You can see in the F1 there is, okay, in the F1 you don't have any recessive phenotype. Phenotype is physical appearance. In the F1, all the children they are going to be tall because they all have a capital T. Okay, recessive phenotype disappears in the F1. This is true, right? Okay, in the F1 recessive phenotype disappears. And then B, dominant phenotype disappears in the F2. Look at that. In the F2, you still have something that is dominant. Look at F2, you have TT. TT, actually, there are three. Okay, so this is false. Recessive phenotype reappears in the F1. It doesn't reappear, it starts disappears. Recessive phenotype appears in F1. No, there is no. Codominance phenotype disappears. There's no even codominance here. Okay. In a monohybrid test cross, remember this is a test cross. In a monohybrid test cross, all the progeny have the recessive phenotype. You can just use this one. Okay. Remember what happens in a test cross? In a test cross, You are using a recessive phenotype, something that is recessive. You are crossing with something that is dominant. It can either be homozygous. If it is homozygous, it means that all the offsprings are going to have a capital T and a small t. You can try to cross that. But if one of the alleles is a small t, but you are crossing with only small t because that's a test cross, this means that you are going to have two capital T and small t, okay? And then you are going to have two small t, small t. So all the progeny have the recessive phenotype. This is false because not all of them have got recessive. The progeny phenotype is 3 to 1. This is also not true. There's no 3 to 1 here. Progeny phenotype is 1 to 1. You can see for this one, you have got half tall and half short. That is a 1 to 1. Okay. The progeny phenotype is 1 to 2 to 1. No, this doesn't appear here unless in a normal monohybrid. The progeny have three phenotypes. Phenotypes are the physical characteristics. Like if this guy was also tall, you only have one phenotype. You only have tall. But if this one was actually recessive, you can see I've got two phenotypes. So I've got tall and short. Okay, phenotype is a physical appearance, so that is also false. So you can say the best option is option 3. A monohybrid test cross is used to determine the phenotype condition of a dominant parent. Okay, this is true. Remember, you are actually trying to determine the phenotype of a parent. Okay, and then test the genotype. You don't test the genotype. Okay, this one is false because you are not testing the phenotype but the genotype. So test the genotype. You already know the phenotype if someone is tall or someone is what. Test the genotype condition of a dominant phenotype. Okay, you test yes the genotype condition of a dominant phenotype. This is true. Test the genotype condition of homozygous. No, you don't know if it's homozygous or heterozygous. Even this is false because of heterozygous. Test for both genotype and phenotype. This is also false. One pure variety of peas has yellow seed which have a smooth shape, while another. Okay, this one here. Anyway, we can try to answer. One pure variety of peas has yellow seed which have a small shape, while the another variety has green seed which have a wrinkle shape. F2 generation of these varieties they've shown. Now this one is a dihybrid. We'll answer it in the next class. Okay, even this one. Dihybrid. Choose a statement which is false. An arrow is responsible for many visible traits. Okay, this is actually true. Every trait is potentially affected by many genes. Okay, I'll just put a dot there. The sequence of nucleotides in a gene specifies the sequence of amino acids. The sequence of nucleotides. Yes, this is to remember this is in mRNA transcription. Every amino acid is coded for by one codon. This is true. Many traits are affected by both the environment and gene. This is true. The reason as to why you can be tall is not just because of your genetics, also the environment, like how are you eating, all right? So every trait is potentially affected by main gene. This is false. Each gene will only give you one trait. Let's say, for example, a gene for eye color, a gene for height. Study the homologous chromosome. We already answered this, and we already did this. The idea that different pairs of full alleles are passed to offspring independently is Mendel's principle of. We are calling that as independent assortment. We'll talk about a lithogen later. From a cross of heterozygotes, which of the following ratios indicate presence of lithogen? We'll talk about also a lithogen later. Talk about that later. Someone who is homozygous has only one dominant allele for a gene, really not. Only one recessive allele, no. Two different alleles, no. Neither dominant nor recessive, no. Two copies of the same allele, yes. Okay, it's two copies of the same allele. Answer this one also later. An attached air lobes is dominant to attached. So remember, an attached is dominant. Let's try to use capital R for dominant, and then the attached is recessive. The attached is recessive, we can use a small r. And then the question is, two parents both with an attached. So you can see both have got an attached, meaning that both of them have got a capital R. Are the child with attached? So they are the child that has got attached. What are the chances that their next child will have attached ear lobe? So the only way someone that has got a dominant allele can have someone that has recessive, it means that both parents are capital R, small r. So that when you cross them, this is going to be capital R, capital R. This is going to be, the other one is going to cross there. This is going to be like this, capital R, small r. And this is going to be capital R, small r. This is going to be small r, small r. So you can see the only way someone can have unattached earlobes is 1 out of 4. If you multiply that by 100%, that is 25%. So there is 25% that the next child can have an attached earlobes. Okay, this one is dihybrid. We'll answer it also. Okay, this is monohybrid. A cross between plants with red flowers were crossed. So capital R is red. Were crossed with plants which bear white flowers. Okay. The offspring are the pink flowers. Now remember, you are crossing something with red with something that is white, and then you're having pink flowers. Use the information to answer the question. Identify the parental cross which gives rise to the results. Now, I think they made a mistake here. So for red, it's supposed to be C, R. For white, it's supposed to be C with a W. You can see on the options there is a W. So you have, you can do the cross. You can either use a double or you can simply use the direct one. So there is a C with R, and then another C with R. Okay. And then there is a C with W, another C with W. So when you cross this with that, to be CR, CW, this with this, CR, CW, this with this also CR, CW, this with this is also CR, CW. Okay. We've been told that the offspring are the pink flowers. So meaning that all of them are a combination. Okay, all of them. So we are crossing CR, CR, and CW, CW. It's this one. Identify the kind of inheritance. If you are getting a color that is in between, you add red and white, and then you are getting pink. That is incomplete dominance. From a cross of heterozygotes, which of the following ratios shows codominance? Now these are hetero. Let's say you have got capital R, small r, capital R, small r. When this guy crosses with this guy, you are going to have capital R, capital R. This one crosses this one, you are going to have capital R, small r. This one with this one, capital R, small r. This one with this one, small r, small r. So this one, let's say the R is representing color. They say R is capital red, the capital is red. So meaning that this one, when we have got capital, capital, it is going to be red. Let's say when you have got small r, that is blue, this is going to be blue. But because this is a codominance, okay, when it is codominance, as long as you have got a mixture of the capital R and the small r, then you are going to have a mixture of red and blue, so it's going to be red and blue. So which of the following ratio shows codominance? So in codominance, there's going to be a ratio. You have got 1 which is red, and then you have got 2 which are red and white, and then you have got 1 that is blue. So it is 1 to 2 to 1. I meant red and blue, not red and white. What type of blood group will the offspring of the following parents have? A mother has blood group AB, A and B. The father with blood group O. Now you can only have blood group O if both your antigens are O. That's OO. So meaning this will cross with this, that is going to be OO. This with that, OO, and then this with this, BO. This with this, BO. As long as you have got O, it means it's going to it's all antigen is always recessive. So these two are blood group A, these two are blood group B. So the only blood group that is possible is A or B. This is a dihybrid which we'll consider in the next class, and this is where we are going to end with today's class. Thank you so much for being present.